Dear students, after studying this module, you shall be able to know about the three important coenzymes, that is nicotinamide nucleotides, flavin nucleotides and lipoic acid. You should also be knowing about the mechanism of oxido reduction by NAD plus and NADP plus. You will also be able to understand NADH and NADPH dependent reactions and learn about the metabolic role of coenzymes. Now students, let us begin with the introduction of this topic, coenzyme. This group consists of three important coenzymes, that is group of coenzyme II, all of which assist different enzymes in oxidation reduction reactions. And this group comprises of first nicotinamide nucleotides, flavin nucleotides and lipoic acid. The dehydrogenation of an alcohol to ketone or an aldehyde is one of the most frequent biological oxidation reduction reactions. As you can see here, we have simplified this reaction and presented to you here. Although the hydrogen atoms removed from substrate are often indicated simply as 2H, it was recognized early in the 20th century that they are actually transferred to hydrogen carrying coenzymes such as NAD+, NADP+, FAD+, FMN, lipoic acid, etc. We shall be telling you about the details about the abbreviated symbols like NAD+, NADP+, FAD+ and FMN later on in this module. They may be considered as carriers of hydrogen or carriers of electrons in metabolic reactions. When NAD+ becomes reduced by dehydrogenation of alcohol, one of the hydrogen atoms removed from alcohol becomes firmly attached to NAD+. Converting it to NADH, the other is released as a proton. As you can see here, the proton which is released gets attached to NAD+, and this is shown here clearly in this figure. Why are there four major hydrogen transfer coenzymes? That is NAD+, NADP+, FAD and riboflavin phosphate, which is denoted or abbreviated as FMN instead of just one. Part of the answer to this question is that reduced pyridine nucleotides like NADPH and NADH are more powerful reducing agents than are reduced flavins. Conversely, flavin coenzymes are more powerful oxidizing agents than NAD+ and NADP+. This difference reflects the chemical difference between the vitamins riboflavin and nicotinamide, which form the oxidation reduction centers of various coenzymes. Another difference, students, is that NAD+ and NADP+ tend to be present in free forms within cells, diffusing from a site of one coenzyme or one enzyme to a site on another. These coenzymes are sometimes tightly bound, but flavin coenzymes are usually firmly bound to proteins, fixed and unable to move. Thus, they need to accept hydrogen atoms from one substrate and to pass them to a second substrate while attached to a single enzyme. Now let us begin with the discussion of nicotinamide nucleotides in this particular module. These coenzymes are derived from vitamin niacin, or known as commonly to us as vitamin B3. Niacin is synonymous with nicotinic acid. You can see here in this figure the structure of nicotinic acid. NAD+ and NADP+ are the active forms of niacin. They are called nicotinamide adenine dinucleotide, that is NAD, and nicotinamide adenine dinucleotide phosphate, which is NADP respectively. Collectively, they are called pyridine nucleotides. And you can see here the structure of NAD and NADP. NAD+ is a dinucleotide made up of two nucleotides joined by a phosphodiester bond. One nucleotide is made up of nicotinamide, that is in the N base, with ribose and phosphate. The second is AMP. In NADP+, the second carbon hydroxyl group of ribose of AMP is phosphorylated to form NADP nicotinamide. Nicotinic acid and tryptophan are all precursors of synthesis of nicotinamide mononucleotide, that is NMN, which is then converted to NAD+ and NADP+. In the oxidized form, the nitrogen atom of nicotinamide is positively charged and is written as NAD+ and NADP+. In the reduced form, this charge is, however, neutralized. So, they are in the form of NADH and NADPH. Mechanism of oxido reduction by NAD+ and NADP+. Now students, let us focus our attention to this particular mechanism. Two hydrogen atoms are transferred from the substrate of NAD+ or NADP+. One hydrogen atom is transferred as hydride ion, that is H- to pyridine ring to nicotinamide whose positive charge is neutralized. The other proton goes free in medium as a byproduct. So only one hydrogen atom is attached to the pyridine ring. That is why reduced NAD+ is always written as NADH plus H+, not as NADH2. So you can clearly see this mechanism here in the figure as given to you here. Why are these two pyridine nucleotides NAD+ or NADP+ differing only in presence or absence of an extra phosphate group? One important answer to this question is that they are members of two different oxidation reduction systems, both based on nicotinamide but functionally independent. The experimentally measured ratio of NAD+ concentration and NADH is much higher than the ratio of NADP+ concentration is to NADPH concentration. Thus, these two coenzyme systems can operate within a cell at different redox potentials. A related generalization that holds much of time is that NAD+ is usually involved in pathways of catabolism, where it functions as an oxidant while NADPH is often used as reducing agent in biosynthetic processes. Typically, NADPH is said to participate in reductive biosynthesis. Now here in this table, we show to you examples of NADP+ and NAD+ dependent enzymes. Let us first concentrate on NAD+ dependent enzymes. First one is glyceraldehyde 3-phosphate dehydrogenase. Second is lactate dehydrogenase. Third one is alpha ketoglutarate dehydrogenase. And fourth and the last is beta hydroxyacyl coenzyme A dehydrogenase. However, on the other hand, NADP+ dependent reactions or enzymes are glucose 6-phosphate dehydrogenase, glutamate dehydrogenase and 6-phosphogluconate dehydrogenase. Some examples of NAD+, NADH or NADP+, NADPH dependent reactions are shown to you here. First one you can see here in the figure is formation of 1,3-bisphosphoglycerate, where NAD is being converted into NADH. However, the second one is the conversion of glucose 6-phosphate to 6-phosphogluconate. Again here, as you can see students in the figure, NADP is converted into NADPH. However, in the formation of 1,3-bisphosphoglycerate as in the earlier example, NAD+ was getting protonated to form NADH. NADH dependent reaction. NADH dependent reactions are usually reductions in the biosynthetic reactions. Hence, these are called as reductive biosynthetic reactions. NADPH dependent reactions. Beta ketoacyl coenzyme A dehydrogenase in fatty acid biosynthesis. Second is HMG coenzyme A reductase in cholesterol synthesis. Third is the phenylalanine hydroxylase for conversion of phenylalanine to tyrosine. And fourth one is the dihydrofolate reductase for synthesis of tetrahydrofolate from folic acid. Now let us talk about coenzyme FMN and FAD. Flavin nucleotides are derived from vitamin B2, also known as riboflavin, and are actively involved in hydrogen transfer reactions. Here shown to you in the figure is a structure of FAD and FMN. Riboflavin is a 6,7-dimethyl isoalloxane bound to ribitol, alcohol of the ribose, that is the sugar ribose. The intense yellow color is due to isoalloxane ring. Active coenzyme forms are FMN and FAD. FMN, which is flavin mononucleotide, and FAD, which is flavin adenine dinucleotide, are active or coenzyme forms of riboflavin. It is shown here clearly in the figure that what are the structures, what are the basic components of FMN, AMP, which compose FAD. FMN is formed by phosphorylation of riboflavin by kinase using ATP in the intestine. This reaction is clearly shown to you here, that using the energy of the cell, that is the ATP, adenosine triphosphate, riboflavin is converted to FMN by its phosphorylation. Now what is the metabolic role of coenzyme? Riboflavin containing proteins are called flavoproteins. Many oxidoreductase enzymes are flavoproteins containing FMN and FAD as prosthetic group. Examples of L-amino oxidase, FMN dependent, they are succinate hydrogenase in citric acid cycle is FAD plus dependent. These enzymes participate in reversible oxidation reduction reactions. Nitrogen atoms at position 1 and 10 in isoalloxane ring of riboflavin undergo oxidation reduction. Example of enzymes requiring FMN or FAD as a coenzyme and reactions where they are involved. Now let us study that. Flavoprotein enzymes are amino acid oxidases, xanthine oxidases, succinate dehydrogenases, acyl CoA coenzyme A dehydrogenase, NADH dehydrogenase and pyruvate dehydrogenase, as well as alpha ketoglutarate dehydrogenase. The pathway involved or the reaction involved in case of amino acid oxidase is the deamination of amino acid. For xanthine oxidase, purine degradation. For succinate dehydrogenase, citric acid cycle. For acyl CoA dehydrogenase, it's fatty acid oxidation. For NADH dehydrogenase, it is respiratory chain in mitochondria. And for pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase, it is oxidative decarboxylation of pyruvate and alpha ketoglutarate in the TCA cycle. FMN dependent enzymes. L-amino acid oxidases, that is the first category in FMN dependent enzymes. These L-amino oxidases catalyze oxidative deamination of amino acid producing hydrogen peroxide, which is split by the enzyme catalase. Here we have given to you the reaction which is involved in the same. Second in this category is NADH dehydrogenase complex. This enzyme is a part of electron transport chain and contains FMN. The electrons are transferred from NADH to FMN and then to coenzyme Q in the electron transport chain. Reactions of FAD dependent enzyme. They involve the reduction of succinate to form a unsaturated diacid. Also they involve the conversion of glycerol 3-phosphate to dihydroxyacetone phosphate, the conversion of pyruvate to acetyl coenzyme A and of alpha ketoglutarate to succinyl coenzyme A. Next students, the topic for discussion is lipoic acid. Lipoic acid acts in the transfer of hydrogen during oxidative decarboxylation reactions. As you can see here in the figure, the structure of lipoic acid, which is a 6,8-dithiooctanoic acid. It occurs both in oxidized and reduced forms as shown here. The oxidized lipoic acid form, as you can see here, it is in the cyclic SS bond form. However, the reduced lipoic acid is in the open chain SH-SH bond form. Also, the given figure shows the structure of lipoamide, where lipoic acid is bound in the amide linkage to the eta amino group of lysine residue of dehydrolipomide acyl transferases. The complex reactions of the carbohydrate metabolism catalyzed by pyruvate dehydrogenase system and L-ketoglutarate dehydrogenase require participation of lipoic acid. It acts as a carrier and undergoes interconversion between reduced and oxidized form. Like you can see here the structure of lipomide. The two thiol groups that can undergo reversible oxidation to a disulfide linkage, similar to that between the two cysteine residues in a protein. Because of its capacity to undergo oxidation reduction reactions, lipoate can serve both as electron hydrogen carrier and as an acyl carrier. And here you can see acetylated form of the lipoate. Lipoic acid is also known as alpha lipoic acid or ALA and thioctic acid. Lipoic acid is cofactor for at least 5 enzyme systems. 2 of these are in the citric acid cycle or the TCA cycle, through which many organisms turn nutrients into usable energy. Example is the pyruvate dehydrogenase complex, the alpha ketoglutarate dehydrogenase, the branch chain oxo acid dehydrogenase, that is BCDN complex, acetoin dehydrogenase complex also. The most studied of these is the pyruvate dehydrogenase complex as shown to you in the figure. These complex have 3 central subunits: E1 pyruvate dehydrogenase, E2 dihydrolipoyl transacetylase, E3 dihydrolipoyl dehydrogenase, that is E3 again. These complex have central E2 core and other subunits surround this core to form this complex. In the gap between these two subunits, the lipoyl domain ferries intermediates between active site. The lipoyl domain itself is attached to a flexible linker to E2 core. Now students, after having studied different types of coenzymes in the coenzyme II section or the category, let us summarize this particular module. 3 important coenzymes which assist in oxidation reduction are nicotinamide nucleotides, flavin nucleotides and lipoic acid, and these are the 3 which we have concentrated upon in this particular module. Further, we have taught you that riboflavin is a component of FMN and FAD, whereas niacin is present in NAD and NADP. Lipoic acid is also known as ALA or alpha lipoic acid or thioctic acid. Further, we concentrated on the fact that NAD plus is a dinucleotide made up of 2 nucleotides joined by phosphodiester bond. One nucleotide is made up of nicotinamide N base with ribose and phosphate, and second is AMP. NAD plus, the second carbon hydroxyl group of ribose of AMP, is phosphorylated to form NADP plus. 2 hydrogen atoms are transferred from substrate to NAD plus or NADP plus, and 1 hydrogen atom is transferred as hydride ion, that is H minus, to pyridine ring to the nicotinamide. The other proton goes free in the medium as a proton, that is H plus, and reduced form of NAD plus and NADP plus is written as NADH plus a proton or NADPH plus a proton. NAD plus is usually used in the pathway of catabolism as an oxidant. NADPH is often used as a reducing agent in many biosynthetic processes, as we have discussed clearly in this module. FAD and FMN have isoalloxone ring which accepts hydrogen ions or hydrogen from substrates and converts itself to FADH2 and FMNH2. NAD plus, FAD plus, FMN, NAD plus, NADP plus and lipoic acid serve as coenzymes for oxido-reductase enzymes involved in carbohydrate, protein, lipid, nucleic acid metabolism and electron transport chain. Hope so students that you found this module very informative.